Our last speaker is Arturo Casadevall, who is just on the slide there. Arturo is a Bloomberg Distinguished Professor and Chair of the W. Harry Feinstein Department of Molecular Microbiology and Immunology at the Johns Hopkins Bloomberg School of Public Health. The major focus of his work has been Cryptococcus neoformans. He's made numerous contributions, including uncovering strategies of latency, treatment, resistance, and dissemination by the intracellular pathogen. He has described the capsule organization and microstructure of melanin and showed how antifungal antibodies mediate immunity against intracellular infection. Dr. Casadevall has been elected to the AAAS Fellowship. He's a member of ASCI, the American Association of Physicians, the American Academy of Microbiology, and the National Academy of Medicine. So we're really pleased to have Arturo come and speak with us. He's going to talk to us about his glimpse of the future. Thank you, Arturo.
So thank you, Pete, for the very nice introduction. Thank you for inviting me. As Joe was talking, I kept thinking that 41 years ago, this place serves as a venue for probably one of the most important scientific conferences in biology ever. And that led to the pause, and that led to figuring out how to go forward. And it has given us the molecular biology revolution. So I think that it's important to reflect on that as we're here. The other thing I want to say is that I had the easiest talk of the evening. I have no idea what's going to happen in the future. On the other hand, it's the hardest one because you can't get up here and do what is done in politics, which is basically say whatever you want and not fact check it or anything like that. I'm going to have to try to be somewhat reasonable. So I want to begin with one of the most important American philosophers of the 20th century, which was Yogi Berra. He was a baseball player, for those of you who are not Americans, and he used to say things like this: it is tough making predictions, especially about the future. But I want to begin the talk actually where Jack was covering. I want to talk a little bit about the past first. And I want to point out that at the beginning of the 20th century, at the end of the end of Victoria's reign, when 7 of the mycoses were found, finding a case of disseminated fungal disease was extremely rare, period. This was a large immunocompetent population, and these diseases, some of them have gone away, some of them are still with us, but certainly you would have not been very hard-pressed to find one. However, 100 years later, the now fungal diseases are distressingly common, and the question is, what happened? What happened is that the hosts were changed. And this was changed by medical progress. A lot of things that you could do at the end of the century, you could save lives in ways that you couldn't do anything at the beginning. For example, IV lines were introduced. Plastics in the 1950s allows IVs to be left on for a while. Immunosuppressants, chemotherapy, ICUs, the cataclysm of AIDS, such that by the beginning of the 21st century, cryptococcosis was eclipsing tuberculosis and competing with childhood diseases as the leading killer in sub-Saharan Africa. So I took my charge seriously. I was told to talk about the future, and I'll tell you what I think is going to happen, and I don't mind if I'm wrong, but I think that what are my visions? I think we're going to see a lot of new fungal diseases. I think we're going to be embracing the x-axis. We're going to be enhancing the symbiont. There is going to be a renewed focus on the host and continued increasing importance for medical mycology. So first, let's talk about new fungal diseases. These are diseases that I believe will happen in this century, and we will basically confront problems that we have no experience with. So people fear viral diseases, bacterial and parasitic, but nobody seems to be afraid of the fungi. People worry about Zika virus, they travel and they take malaria prophylaxis, they worry about tuberculosis, they worry about getting meningococcal meningitis. You don't see anybody, the general public. And what is that? Well, it's very interesting because we tend to be relatively invulnerable to this. Fungi are currently devastating entire ecosystems, and often a single species can wipe out and lead to the extinction of some of these representatives up there. In contrast to communicable diseases such as the bacteria and the viruses that are very dependent on the host for their survival, fungal diseases do not ultimately need the host. They will kill every single individual in a species, and they're the only ones that can reliably be counted on to cause extinction. So, what is this resistant from? Well, some of the work that has been very influential in my thinking came from John Perfect's lab. And this is work that was carried out in the 70s, in early 80s. And what he showed basically is you take Cryptococcus, you take a bunny, and it's very difficult to give that bunny disease with Cryptococcus. It will clear it, even if you put it directly into the brain. So you can, however, put it in the skin. The skin is cooler. If you, however, give steroids, you end up with a dead bunny. So it begins to suggest that what you have here is the emerging pattern is a combination of endothermy and adaptive immunity. You see an example of the importance of temperature in the devastation of the bat population by white nose syndrome. So bats in the summer have the same temperature than you do, 37 degrees, and they're pretty much impervious to this organism. During the winter, as insects disappear in the northern hemisphere and they begin to hibernate, their temperature drops to 10 to 12 degrees. And when that happens, this fungus essentially grows on them, invades them, and they wakes them up in the middle of the winter. There is no food, and they die. And these caves are full of dead bats in the middle of winter. So, but raising temperature alone can cure fungal diseases. If you take those bats that are heavily infected and you bring them into the laboratory and just feed them, they get their metabolic rate up, they will be able to be cured of white nose syndrome. Frogs that are infected with a chytrid, if you take them from the lake and you put them in a warm room at 37 degrees, they will be cured. And for insects and ectothermic vertebrates, induced fevers will increase survival. So some years ago, Vincent Robert and I carried out a study in which we analyzed the CBS collection. So it turns out that when a sample is submitted to the CBS collection, they do a pretty careful temperature susceptibility study. And there were 4,000 strains there, and what you can see is that there is a very rapid, I thought it was a green one. Did you have a green one, Joe? No? What? Oh, okay. Yeah, it's a lot better. So what you can see is that, most fungi do well in ambient temperatures. Thus, they live in the environment. However, as you begin to raise the temperature, there is a precipitous drop in the percentage of them that can replicate at that. In fact, for every degree that you increase, you can exclude 6% of fungal species. That's a tremendous amount of protection that you get with temperature. So we took that data, which is the benefit. So approximately 6% for every degree Celsius. And what is the cost of being had? The cost of being had comes from an equation that basically is pretty much dependent on the temperature. The hotter you are, the more you need to burn calories. And these two differential equations were solved simultaneously, and this was the maximum. The maximum is actually you're very close to your temperature. Now, this could be coincidence, but again, it wasn't 28 and it wasn't 46. It was the temperature, and it does begin to make sense. Now, so many of you have heard me argue that, in fact, we're here because a fungal filter is selected from mammals or reptiles. So you have the age of reptiles, you have the KT boundary, where there was a cataclysm followed by a global compost, and the disease situation with massive fungal proliferation could have selected for those individuals that were warmer at the time. Mammals make no sense, period. Mammals need to eat a lot than any of the other animals on the planet. They reproduce relatively slowly. They spend time caring of their young. There is no really good explanation why we did not have a second reptilian age. But I believe that you could argue that fungal selection created a filter for the emergence of mammals, and this has been laid out if you're interested in an essay in PLOS Pathogens. So what's going to happen this century? Well, this century, first of all, if you look at the major mycoses, they are near the equator. That's consistent with organisms that are more adapted to higher temperatures. The planet is going to get very warm. And depending on and yes, you know, this year is already the warmest year ever. Last year was the warmest year previously. So what is going to happen is that as the temperature rises, the difference between your temperature and the average environmental temperature is going to get smaller. So that window that is preventing us from fungal diseases is going to get smaller. Now, fungi can be very rapidly adapted to live at higher temperatures. This is an experiment that was done by a company in Florida. And what they did is they took an organism, a fungus that they wanted to use for insect control, but the insects learned to sun themselves. So the insects could cure themselves of this agent. So what they did is, within 6 months, they managed to get it to grow at 37 degrees. This is not that hard to do. These organisms are very pliable. And so as the temperature rises, you're going to have more of these organisms probably being able to meet and grow in our temperatures. Now, we tried to ask the question a couple of years ago, this was published last year, as to what is the story with thermotolerance in fungi. By thermotolerance here, we're talking about the ability to grow at 37 degrees. So first thing you notice is that it's not monophyletic. The second thing you notice is that there are a lot more ascomyces that are thermotolerant than basidiomyces. And that's consistent with the overwhelming majority of human pathogenic fungi being ascomyces. So it correlates with latitude. The higher you are, the less thermal tolerance. And it correlates with the capacity for fermentation, and that makes a lot of sense. Fermentation generates a lot of heat. When you, however, begin to look at this, you see tremendous complexity, such that high and low tolerance appear to be discontinuous, suggesting possibly different mechanisms by which these organisms survive at the extremes of the temperature range. Thermal tolerance correlated with the ability to ferment certain sugars and some other chemistry. So I think the most important thing, however, was at least what comes out of this is an explanation for the fact that ascomyces are our primary fungal pathogens. But here is something that a reviewer asked us to do, and it was a great suggestion, which was to take the data at which the colonies have been deposited in the collection and to plot it for thermal tolerance. And over here you can see the ascomyces. The ascomyces are already on average, temperature tolerance, so they don't change very much. But look what's happening with the basidiomyces. And they're rising very, very rapidly. So obviously this is only suggestive data. You shouldn't go from a correlation to making an association, but it does suggest that these organisms are in fact already adapting, and that we are going to be seeing some of them break through to become mammalian pathogens. So the alternate virulence hypothesis, by the way, I noticed that this alt that is now being associated with a political term is already in the lexicon. It's used for like alt Latin, food, alt Latin. So I am playing with it. I'm not sure that I'm going to use it, but I'm trying it out on you, see what happens. So it posits that the capacity for microbial virulence emerges by alternative selection forces independent of the final host. These selection forces involve both by abiotic, temperature, humidity, etc., and biotic amoeba causes. It views the emergence of virulence as a continuous stochastic process, and for fungal pathogens, there is now considerable evidence that grazing by amoeboid predators select for traits that confer upon the microbe the capacity for virulence. I'll show you some unpublished data from the laboratory of Alan Guimarães in Brazil. So what you're seeing here are just the ability of amoeba to eat all these different pathogenic fungi. Here, however, the most interesting data, he basically let the amoeba select for it, and look, the amoeba ended up increasing the virulence of all these organisms, Paracocci, Histo, forgot what that is, Cryptococcus, Candida, and even Saccharomyces cerevisiae. So the next thing he did was he began to look for the receptor, and it appears that the receptor is this protein, which is about 45 kilodaltons. And we're very interested in this because this receptor would be able to take in a encapsulated Cryptococcus, which has a very different surface than a non-encapsulated Cryptococcus, and it may give us some hints as to what are the features that are used for recognizing all these polysaccharides. Now, amoeba and other fungi, we looked at Sporotrix, Blasto, Histo a long time ago. We looked at Crypto too, that's not in the slide. Other groups have looked at the entomogenous fungi, that is, the idea that even the fungal pathogens of insects also get selected by this mechanism. Patricia and Ildinet laboratories have similar data with Paracocci, and there have been a couple of papers for Aspergillus. So virulence then emerges from selective pressures in the soils that are independent of the final host. And one way I began to think about this was that, in fact, why only few of these organisms, why are only a few of these organisms pathogenic? Why you don't end up with all of them or most of them? Well, imagine they're all playing a game of cards, and imagine what the things you call virulence factors are different cards. You could have a card for thermotolerance, urease, phospholipase. You could have a molecule that can function as an adhesive, and these essentially all of them get shuffled. Well, if you do that, you could see that a mammalian hand always has to have growth at 37 degrees. Plant hands don't need this card. They may need other attributes in order to break down plant matter. Insect hands will also not need that one. And I actually thought that this was kind of pretty clever until I realized that somebody had at least thought through this possibility before. So just to summarize, the bottom line is that virulence can emerge independent of the final host, and that it emerges stochastically, and that probably selection pressures are out there are already selecting organisms with the capacity of it. So the person who had thought about this was H.G. Wells, War of the Worlds, because the Martians had never been to Earth, so they landed in Earth in the 1890s. They beat up the London Police Department, and within a few days, they all succumbed. They had the bad luck to land on a very dirty planet. And even though none of these organisms had actually seen a Martian before, some of these organisms had the capacity to bind to Martian adhesins and to mess up their immune system. It's so, at least if you think through it, this story was there already. So some predictions: new fungal diseases will appear. The first emergence will be in warmer climates. The first evidence may come in cooler mammals. What are cooler mammals? Things like the platypus, which, by the way, at the maximum temperature is 31 to 32, and they are very susceptible to fungal diseases. We're going to see more pathogenic basidiomyces, and some may be resistant to all antifungals. So can we do anything to prepare? Well, we know very little about the fungal world out there. Certainly very few of these organisms have been studied. But to assess the threat will require greater knowledge of fungal biology, ecology, and thermosusceptibility. But I would argue that even if you did that, it would be very, very difficult, if not impossible, to predict which will make the jump. So this is an experiment that we did a few years ago, in which we took 3 cryptococcal species, And you can see that two of them are not pathogenic for humans because they cannot grow in human temperatures. So you had to you couldn't do this experiment in mouse. You had to go to a host that could be at lower temperatures. And here you have Cryptococcus neoformans with some of the major virulence factors like capsule, melanin, phospholipase, and urease. And this organism here has them too. But it turned out that the one that was the most virulent didn't have the capsule, and it did not make melanin, at least not in these conditions. Suggesting, in fact, that it may be that if you know everything about a host, and if you know everything about a microbe, and you can imagine them mixed together, you may be able to take a guess of whether there'll be some pathogenic potential. But I don't believe that microbial pathogenesis will ever be a fully predictive science, although it may be like the weather. You may be able to say this combination of microbe and host has some probability. So next, my vision is the embracement of the x-axis. There has been little progress in treating fungal diseases in a generation with regards to reducing morbidity and mortality. The drugs are not enough. So what am I talking about? You basically move in this direction. This entire axis has been ignored by infectious disease. But think about what would happen if you could enhance immunity, just move on it. You could take this host microbe interaction that is in trouble over here and move it over here to be beneficial. Or if you were at the other end, you could reduce immunity. You could also do the case. There's a tremendous therapeutic potential on this axis. Let's think about tuberculosis. You have miliary disseminated TB. There is evidence that if you add gamma interferon, you move in this direction. Or you could have TB meningitis. Sorry about that. I made that slide about 10 minutes before dinner. That's how this talk went to the wire. And you could add steroids and you can move in this direction. And there is some evidence, for example, that this does happen. There is some individuals who treat some aspects of cryptococcosis when they use steroids. Tom Harrison has done beautiful work showing that there is some benefit to the administration of gamma interferon. The problem is that today, when you go see a patient at the bedside, you don't know where they are on this curve. So because you don't know where they are on this curve, it is very difficult to add immunomodulation. Now, if you look, mycotic diseases occur at both horns of the parabola. So here you have cryptococcosis, you have disseminated disease in this end, you could have iris, cryptococoma, there is some evidence for asthma. You could do this for aspergillus. So you have disseminated aspergillosis here, bronchopulmonary aspergillosis, asthma sinusitis at the other end. You can do it for histoplasma. You can have disseminated histoplasmosis at this end and mediastinal fibrosis at this end. Both of them will put you in the grave. The way there will be different, but they're just as lethal. And for Candida, I was very pleased to see that Mary Risk, together with a couple of other people in the Candida field, have taken the damage framework and adapted it. And what they did is they've broken the parabola into its constituent parts and show that in fact vaginal candidiasis is probably all host damage or as the result of host damage from immune system, while oropharyngeal candidiasis you have damage in the absence of immunity, probably from the massive amounts of organisms. So some predictions, technology will become available that would allow physicians to determine where the patient is in the damage framework curve. The therapy would evolve from kill the microbe to help the host. Inflammation would be titrated for maximal antifungal efficacy and minimal host damage, and fungal diseases will be treated with combinations of antifungal therapy and immunotherapy, enhancing the symbiont, buttressing the internal defenses that we already have to reduce fungal diseases. So the first thing I want to tell you is, again, this is the curve that you saw before. You can move along this curve. Now, it turns out that fever, people have been trying to establish a beneficial role for fever in vertebrates, in us, for decades. And you can find studies for and against, and you could find arguments for and against. It's very easy to show a role for fever in, for example, ectothermic organisms. You take a lizard, you give it a fungal infection, you don't let it sun itself, this lizard will die. The same thing with a... So what is different about humans versus... Why has it been so hard to establish a role for fever in humans? Well, this is a paper that appeared early this year in which what I argue is that this has been in front of us all along. Most diseases that physicians treat in the hospital are caused by microbes that are already thermally adapted. Your staph, your E. coli, your candida, for example, are thermally adapted. So it is going to be very hard to show 2 or 3 degree difference having a therapeutic effect. But what would happen if you were infected with an organism that the maximum temperature was 38? In that situation, a small change in this here could result in a major therapeutic benefit. I want to point out that therapeutic fevers were used up until 1940. They were used for the treatment of syphilis and gonococcal infections. Warner Jarak got the Nobel Prize for inducing fever therapy with malaria, Plasmodium. Hippocrates wrote in antiquity, "Give me the power to produce fever and I will cure all diseases." This is the Kettering hypertherm. So what they will do is, if one had syphilis or gonococcal infections, they will put the individual here. Notice it's got a fan to cool the face. And this will rapidly change, the temperature will rapidly go within half an hour to 41 or 42 degrees. And thousands of patients were treated and safeties were done. And I reviewed this data, and believe it or not, people tolerated it extremely well. There were a couple of cases of seizure in which they went too high, and there were a couple of cases in which thermal burns were done because it was defective on it. But this is a forgotten element of history. And if you had one of these very difficult to treat fungal infections, maybe, and you knew what the temperature susceptibility, you may imagine a diagnosis, take the organism, determine their thermal susceptibility. If it is greater than 41, obviously antifungal therapy, immunotherapy, or you can target temperature. Joe does a lot of work with the calcineurin pathway. It may be possible to target temperature, make these organisms more susceptible to temperature in vivo. But what would happen if the temperature was less than 41? Well, antifungal therapy, immunotherapy, maybe fever therapy will be in the future. Just fry the organism. The microbiome manipulation. So Mahmoud Genoun has become one of the leaders in this area, and he's going to hopefully talk to us about this in a couple of days. It's a work that he did in 2010. So we are in the midst of a microbe revolution, but it's a revolution that is hollow. What do I mean by that? It means that people are sequencing everything, primarily bacteria. Well, it's a lot easier to sequence bacteria and to categorize them. We know very little about the fungal microbiome. And in fact, there is also other eukaryotic organisms there, including the amoeba. So the issue, I believe, is that this is part of what keeps us healthy. And once we learn about it, we may be able to manipulate it to prevent fungal diseases. So I also anticipate there's going to be a focus on susceptible individuals. Fungal infections are extremely common, but disease is rarer. Among susceptible populations, only a few individuals suffer disease. In the time of AIDS, that is, when we used to have people with very low CD4 count, about 10% of the patients had cryptococcosis. People said they have no T cells. 90% of them had no cryptococcosis. There was evidence that they were infected, and they had no T cells. Again, there was a logical error in thinking through that, but it gives you an example that obviously not all hosts are the same. Individual susceptibility is a complex function of genetics, history, microbiota, and immunity, and stratifying risk will make future antifungal therapies more effective. So if you could stratify a risk, this will enable vaccination, immunotherapies, microbiome interventions, and targeted antifungal prophylaxis. In diagnostics, I believe we will continue to make improvements with the current technology. But I also believe that there is a revolution a decade or two ahead of us, and that is the use of imaging to identify fungal infections. I want to acknowledge that Tanya Sorrell and you, Emil Himmelrich, did some of the very early work with using NMR to actually see Cryptococcus in mice in biological fluids. The fungi are very different. So in this future, it may be possible to put the patient in this enhanced MRI, look at the lung, and then just look at the fungal products on the site. And that will give you a signature that would allow diagnosis. And this is beginning to happen. This is a paper from last year, and this paper just came out in PNAS, and you can just download it and take a look at it, but you can see this is going to turn out to be the aspergillus over here, this red stuff. And you can see that you don't see it with Yersinia, strep, pneumo, or the BBS control. So I do think that so the question is, why isn't the technology there yet? The computing power is there. The problem is you got to have bigger magnets, and you got to be able to separate the fungal or microbial signature away from the non-fungal signature. So my last predictions is that medical mycology will evolve into a more host-focused discipline. It has to. It has to. I mean, we basically are pretty much at the limits of what we can do with this existing types of antifungal agents. There will be a gradual but significant improvements in prognosis as microbiologic, genetic, and immunological knowledge is brought to the bedside. Medical mycology will increase in importance relative to where it is now for the simple reason that all medical progress affects homeostasis. And when you affect homeostasis, you're going to end up affecting the capacity of the host to resist fungal diseases, and that's going to translate into an increasing number of them. So with that, I end, and thank you very much.